The overall goal of this procedure is to dissect and image single Drosophila Ommatidia to study intracellular events in neurodegeneration models. This is accomplished by first detaching the fly head from the rest of the body and cutting it open. Next, the brain and other tissues are removed from inside the head. The cuticle is then removed and each retina is cut in half. The final step of the procedure is to carve out the ommatidia with a tungsten needle and deposit them onto a slide. Ultimately, results can be obtained that show cytoplasmic autophagic organelles through fluorescence confocal microscopy.
The main advantage of this technique is that it allows high-resolution cell biological analysis in Drosophila models for neurodegeneration, which until now could only offer genetic insights in this field. Demonstrating the procedure will be Vera Volpi, a graduate student from my laboratory, with the help of Daniel Mackay, a histology and microscopy technician at the center.
To begin, fill 1 well of a 3-well glass slide with phosphate-buffered saline. Then, anesthetize the flies with carbon dioxide. Once the flies are anesthetized, pick up a single fly using forceps and drop it into the small dish of PBS. Under a dissecting scope, pinch the proboscis with forceps and then detach the head from the body by severing the neck using a second set of forceps. Holding the fly head from the proboscis,